UNAM in 2007. He then pursued master's degree in biotechnology at Lund University, Sweden, conducting research in biophysical chemistry under Professor Sarah Linz. In 2014, he completed his PhD in soft matter and physical chemistry at Netherlands under the supervision of Professor Martin Cotton Stewart and Dr. Renko. Following his doctoral study, Dr. Hernández was awarded the prestigious PEW Latin American Fellowship, allowing him to conduct postdoctoral research at the Simpson-Curie Institute for Biotechnology at Northwestern University, USA, under Professor Samuel Stopp. In 2017, he joined UNAM's Institute of Chemistry, where he established the Laboratory of Biomolecular Engineering and Bionanotechnology. His research focused on the design and development of nanostructures, biomaterials, and CRISPR-Cas system for biomedical applications. He is currently level number 2 from the National Research Recognition, is winner of the 2019 Award to Bionano Innovations. So thank you so much, Dr. Armando Hernández, for this talk, and we can start right now.
Well, first of all, thank you very much. For me, it's an honor to be here. In these conditions, at least, I would like to be there, but unfortunately, due to some health issues, it was not easy for me to go there. But hopefully, I will give my presentation in this way, which is perfect because I'm really, really excited to show these results to you. First of all, I would like to say also that I will switch kind of gears because I know most of the talks in this meeting, although they are about sensors, they have different perspectives. This will be very biological oriented, but it keeps the same essence of biosensing, but with biologicals, with enzymes, with these marvelous systems that were given by nature, which are called CRISPR-Cas systems, which we are using for developing various sensors for detecting pathogens. So I will start first just to show a bit about my agenda. So I will start telling you a bit what is CRISPR-Cas systems, because of course it's a very biological topic, and I need to provide some background. Later on, I will show how we are using them for specific applications in like biosensing of pathogens. So let's start. First of all, I have to say that CRISPR-Cas systems in general is like a real molecular Swiss knife system, because I will show you later what is exactly, but these are basically, in a nutshell, an immune system present in bacteria and archaea. And you may ask, well, how it happens that an immune system became sensor? Well, actually, it has a lot of applications. Perhaps you have heard about, because of the chemistry Nobel Prize in 2020, that the CRISPR-Cas systems is mainly used for the development of genome editing. But beyond genome editing, there are many applications, including diagnostics, which is actually one of the first to have commercial application or application massively from CRISPR-Cas systems. But what is exactly a CRISPR-Cas system? As I already mentioned, CRISPR-Cas system is an immune system from bacteria and archaea, which is, let's say, the cousins of bacteria, no? To protect against virus attacks and other attacks from other elements. So CRISPR stands for very convoluted name, cluster regularly interspaced short palindromic repeats, which basically means some sequences present in the genome of this bacteria. So it's an adaptive immune system because it's changing all the time depending the viruses that are attacking the bacteria on archaea. No? So the bacteria capture fragments from the genome from invaders to create a memory, and then it's using this memory later on to fight against future invasions from these viruses or any other elements. But to be effective, this CRISPR arrays, there is a key, a very, very important protein, which is the Cas protein, which makes the full name CRISPR-Cas. So this Cas protein is actually the protein that does everything. So because the genome, well, is useless just like that, but the protein is actually doing everything. So it's recognizing the invader DNA and cutting it. So, and how it happens that it became a sensor, very important sensor and very versatile. So we have a Cas protein depicted here in red. The second element we need is something very, very important called a guide RNA, which is in yellow, which is a RNA molecule, single-stranded nucleic acid type, it works like a GPS because it tells the protein, the Cas protein, where to go. And how it tells where to go to the protein? Because it needs to match complementary the sequence with the target DNA. And that is the way how it recognizes specific sequences of DNA or RNA, depending. There is an extra checkpoint, which is very important, which is a sequence that depends on the protein, the CRISPR-Cas protein. For example, here is 3 nucleotides for Cas9, and the N nucleotide, then 2 guanines. But those needs to be present, adjacent to the sequence that is recognized by the RNA. So this RNA sequence is extremely important because it's programmable. You can change that sequence and you can recognize any other sequence as long as you have this PAM sequence next to it. And it's very specific. So just to tell you a bit of details, I mean, there are many systems of CRISPR-Cas. Here I'm showing those, Cas9, which is the most famous, and Cas12a, which is the one we are using for biosensing. And there are differences. I'm not gonna really go into them, but just to tell them that there are differences. Guide RNA is different. For example, for Cas12 is shorter, for Cas9 is longer. There are these PAM sequences different, et cetera, et cetera. But the important is that the guide RNAs are programmable. So you just need one protein and you can change the guide RNA and you can detect any sequence you want. So we are using the CRISPR-Cas12a system because it has another very important feature, which is, well, we know that because of the guide RNA, it recognizes the complementary sequence, right? And it's find the protein with the RNA, which forms a complex, a tertiary complex with the DNA. And after it recognized completely or almost completely, there are some tolerances, it cuts, it clips the DNA, you know? That is how it, in biology, that is how this helps the system prevent invaders, attack of invaders, cleaving their genome. But it happens that this protein, which is an enzyme with nucleic acid cleavage activity, after cleaving the specific DNA, also can cleave any other DNA that is single-stranded. And this second cleavage state, which is called trans, is exploited to use the CRISPR-Cas systems as biosensing. Because if we have, for example, a short strand of DNA with a quencher of fluorescence of a fluorophore, then once it detects the target sequence, the target DNA, it will cut also this sequence and release a fluorescence. But there are other ways, but this is a very commonly used readout way. And with this, we can detect. So this is the principle of using CRISPR-Cas for biosensing, at least in the most reported methods or the ones that we are using. So we have exploited this to detect various pathogens. For example, SARS-CoV-2, also a parasite called Trypanosoma cruzi, which causes the Chagas disease, very important neglected tropical disease. Also, dengue virus, another tropical kind of neglected disease. And so I'm going to show how we are using this CRISPR-Cas12a system for detecting, and not only detecting, but also for diagnostics. That means already using clinical samples. So everything started in 2012 because it's the first time it was reported, the programmability of CRISPR-Cas systems against a sequence to detect and cleave a sequence. So that is why Jennifer Doudna and Emmanuel Charpentier earned the Nobel because of this paper. So that was in 2012. In 2018, it was reported already CRISPR-Cas to be used as genetic sensors. So a few years, then very immediately, many other reports start to be released. Faster, better, cheaper, the rise of CRISPR in disease detection. And then with the pandemics, early in the pandemics, in April or something like that of 2020, FDA actually gave emergency authorization for the first CRISPR-based diagnostic method for SARS-CoV-2. And then a lot of, news start to appear. Very excited. Many startup companies opened, reporting very impressive analytical sensitivities or clinical sensitivities for detecting, etc. And so you can already actually see why all this excitement was occurred, because I already show you how these CRISPR systems work, and you can see the advantages. And because you only need a protein, right? And you just need to change or to redesign the guide RNAs, which is done quickly in a matter of days, it can be. And the synthesis of RNA is also faster than the synthesis of a protein. And you can synthesize many. But okay, so what is the actual workflow we are using with CRISPR-Cas systems to detect specific genes associated to these pathogens? So we have the aim actually to develop these sensitive, specific, rapid, portable, and low-cost detection methods, which actually is quite ambitious, but I think CRISPR-Cas systems are very well suited for doing this. So what is the workflow? Sorry, if we collect the sample from a patient or any other source, we need to extract the DNA or the genetic material. That is something like, for example, is done for the PCR. After you have extracted or concentrated the genetic material, we submit this to a isothermal amplification. It can be PCR, but also an isothermal amplification. I will talk about more about this later on. And then when we have already enriched the genetic material, we use the CRISPR-Cas system for detecting and generating the signal that will be the readout, for example, in fluorescence, but also in lateral flow strips, fluorescence with a plate reader, naked eye, many other, but those are the ones that we are using. This is the general workflow. So what we specifically did was that everybody was using only one single RNA, not this molecular GPS. So we thought, why not to use multiple RNAs, guide RNAs, against multiple parts of the same gene? Because if you activate more copies of the Cas protein, so the activity will be higher, and probably this will allow to detect the pathogen faster. So we did that. We did this for the Wuhan strain of SARS-CoV-2. The workflow that you already know, I already told you. And so just to clarify, the isothermal amplification we are doing here to enrich the genetic material is a method called LAMP, loop-mediated isothermal amplification. Some people already use this alone, I mean, after the extraction of the material, the genetic material. But the point of this method is very, very sensitive. It can really go, I mean, sensitive to go wrong, or easy to go wrong, or to give opposite answers. So, but we don't care here. We are only using it for enriching. And it doesn't matter if amplify something wrong, the cars will just discern and really detect the proper and correct sequence. So this work done my master's student, Melissa Morales. She designed everything to run this amplification, isothermal amplification with only use one temperature and 10-15 minutes. And then we detect that, for example, combining two different gRNAs, the detection, the fluorescence, because of the cleavage of the reporter probe, this single-stranded DNA, was really, really fast. So it happens only in a few minutes. And when you use this RNA separately, it's slower, corroborating actually our hypothesis. So all this was done in DNA. Actually, the analytical sensitivity was very similar to quantitative PCR, but in lesser time. And actually, we also applied this not only for fluorescence with a plate reader, but also with lateral flow strips. For example, when you here, these strips work that this top line tells you if the test is positive. And so you can see clearly that all here are clearly positive, but only when you combine several guide RNAs when you have this stronger signal. And here the probe is just modified not to have a quencher and a fluorophore, but to have a different system that allows to be captured in these chromatography lateral flow strips. We validate this with samples from patients. So someone that evaluate samples from qPCR gave us, for example, 6 positive samples by qPCR and 5 negative samples. And we detect all the 6 positive as positive and all the negative as negative. Also in the lateral flow strips. You know, all are positive, all the positives are all are negative in all the negatives. And actually the limit of detection is around 10 viral copies, which is this one in red, that still we can detect, which is very, very, I mean, it's an amount of concentration of attomolar. And also, we can use this to detect with naked eye. For example, we just need to increase the concentration of the reporter DNA several times, and then the fluorescence will accumulate. And using a transilluminator blue light plate, we can see here, for example, you can see clearly, this is a positive control, a negative control, clinical samples from a patient which is positive and negative. Because of course here you see greenish, but there's still less because the FAM or the fluorescent molecule is quenched. And well, so we corroborate this actually with environmental samples, other clinical samples, which actually were quite encouraging to continue with this. So we also try to detect or differentiate strains or variants of SARS-CoV-2, no? For example, Wuhan and Omicron. But here we incorporate a novelty. So we thought, what happened if we could just I mean, all these proteins work at 37 degrees, the Cas proteins. But if we could accelerate this, if we increase the temperature, in theory, it was no, because I mean, this protein optimal temperature is 37, 40 degrees. But in reality, what happened is that when we incubate the reaction, I mean, with the guide RNA, etc., and the detection with CRISPR-Cas protein, etc., at different temperatures, you could see how, I mean, all these are positive samples, but you could see clearly how when we use in 65 degrees, the detection accelerate. I'm not showing here the negative controls, but all the negative controls were really flat. So there is no on specificity. We optimize this. I mean, we even increase the speed, changing the buffers, optimizing the buffers, and clearly the sensitivity. For example, at 37 degrees, you could only detect the 5 nanomolar. This is the Cas alone, no isothermal lamp amplification. And with 65, you could go to really to picomolar, to 15 picomolar. But that is without the lamp. With lamp, you go to, as I said before, attomolar. Well, here my student designed guide RNAs depending on the genome of the Omicron and the Wuhan variants to be specific for one variant, but not for the other. And at the end, after testing, we really get guide RNA that could differentiate. For example, this is Omicron at 65, and this is Omicron at 37. So we see the difference because of the temperature. And these two are the Wuhan, which are not detected at all. And also, especially at 65 degrees, we could detect with these lateral flow strips. Another pathogen of interest was Trypanosoma cruzi. So Trypanosoma, well, first, sorry, dengue. Dengue is also a tropical neglected disease. Probably, I'm pretty sure you know that the last year we have quite a lot of number of cases in Latin America, also in Mexico, just 200 cases. It causes a hemorrhagic fever, which can be deadly. So yeah, fast, specific methods for detecting are needed. This work was done by Marco. The interesting part or the challenging part is that there are 4 serotypes of dengue, 1 to 4. So through this pipeline, using different, I mean, all the bioinformatics to do a genetic alignment and detect parts that can be, are commonly shared by all the dengues or only specific to particular serotype of dengues. So Marco designed several guide RNAs and follow the same workflow, the general workflow as I mentioned. And the results show that he could design one gRNA, one guide RNA, that together with the protein Cas12a could detect all the dengue. It doesn't matter if it's dengue serotype 1 or 2, 3, or 4, it could detect it. But also design guide RNAs for specific serotypes, only for dengue 1, 2, etc. And also this was with fluorescence, with a plate reader, but also in lateral flows strips. So for the general detection, only one gRNA for all, or specific gRNAs for specific serotypes. You could see the positive band. And also using some samples from patients. This was from dengue 2, and this is from dengue 4, so we could detect them. So this RNA was extracted from the blood of a patient. Finally, Trypanosoma cruzi, as I was saying, is also a tropical disease, neglected tropical disease, because causes millions of cases every year, thousands of dozens of thousands of deaths yearly, and has a very complex life cycle. So Idalia, my master student doing this project, so follow the same workflow, design from the genome, from the parasite, because it's a parasite, find regions that were interesting to design these guide RNAs. And she found that actually this parasite has something that is called satellite DNA, which is a sequence that is highly repetitive, like thousands of times, and also another sequence from mini circles, kinetoplastic DNA, which is repeated thousands of times. So this was perfect to be a target DNA. So extracting DNA from parasites or even infected mice, I mean, following the same workflow, she could detect by fluorescence. You can see 5 minutes already. It doesn't matter which DNA, the satellite of the kinetoplastic. But here the sensitivity was crazy. The analytical sensitivity was 10 to minus 24 molar, which is joptomolar. This is because, of course, one cell has already thousands of copies of the gene of the target sequence. But this parasite is very complex. There are strains, so we could detect specifically the Mexican strains, these Ninoa and Querétaro, and the others that are in the south. So in South America, no, because everything was designed for the Mexican strains, and also in these lateral strips, and also in infected mice. So finally, I just want to show you quickly about another system that we're starting to use from CRISPR-Cas, which has many more advantages, which is called Cas711. So I was telling you that the Cas12 has the advantage that after detecting the DNA target, has this other enzymatic activity that clips single-stranded DNA that we use. We have a reporter with a fluorophore quencher, etc. And from that, we can detect the presence of the specific DNA because of this signal. This protein, the Cas711, does something similar but different because instead of having DNA reported, it has a protein. I mean, it's a protein that is cleaved. It's not cleaved DNA. So this opens new possibilities. Actually, this is very interesting, the protein, because I mean, it's associated with this CSX29, which is a protease. So a protease associated to CRISPR-Cas. And well, this opens new possibilities because we can redesign proteins to couple to this protease activity associated to detection of the specific RNA sequences to generate cascades, signaling cascades like in the cells, which with a very small amount of initial signaling molecule, we can get a huge effect, a cascade effect, an amplification of signal. So we think we could do something like that with this protein that has a protease. Well, but what we have done, or my student, Gustavo, PhD student, has done so far, I mean, it cuts this Cas711 plus this protease cuts this protein, but how can we recognize the cleavage of this protein? Well, we need to build a reporter. So he genetically fused and produced these proteins coupled to a FRET pair. So a GFP and a red protein, so which is a FRET pair. And so that means when it's cut, the GFP will fluoresces. So, well, he produced all the proteins. I'm not going to show you because the biochemistry of this protein is crazy. But here you can see that in this electrophoresis gel, SDS page gel, so when we have the Cas protein with the protease, we add the reporter protein, this protein that is cleaved next to the FRET pair of proteins, of fluorescent proteins, and also adding the target RNA. This is the target protein, which is cleaved, so it disappears. But this we could see already, so this is very preliminary, in the lysates from bacteria where these proteins are produced. So this is the reporter protein. So you could see a reddish, and this is just for comparison a GFP protein, so without the red protein. So it's clearly yellow, greenish. So the reporter is really doing FRET. And in the lysate, I don't know if you could clearly see, but we could see this is very preliminary. Of course, so when we add the target RNA, the color is less reddish. Here it's not maybe that clear, but it's less reddish, so it's more greenish, yellowish, meaning what I was telling you, that this reporter protein is being cleared. Okay, so just to finish with the conclusion, so I hope I can convince you that CRISPR-Cas systems, these biological immune systems, are very well suited for biosensors, genetic biosensors, using this collateral activity that help us to get sensitivity and specificity comparable to gold standards with qPCR, but faster with portability ways, highly specific sensitivity, and very flexible. And new CRISPR-Cas systems open new perspectives. So just with this to finish, thanking to all my students that participate in these projects and all my lab. I'm open for questions. Thank you very much.
Thank you very much, Professor Armando Hernández. So we have time for 2 questions. Please feel free. Don't be shy for questions. Okay, we have 1 question here.
Thanks a lot for this very good lecture today. And I would like to ask you with respect to the CRISPR-Cas9 detection of the different kinds of viruses which you have, of course, especially COVID-19. So you have been using lateral flow strips, which is, and you have shown that you're trying to detect at 45 minutes, right? So I just wanted to ask you, are you planning to, or is there any strategy wherein we can decrease this time line further? Is there any strategy to understand? Because here it's like 25, 60, it's great because in comparison to PCR, this is really working fine. But do we have any other strategy using this CRISPR-Cas9 that we can decrease the time limit?
Yeah, exactly. We want to reduce the time having these 2 steps in just one. So now we cannot do that because this step is done at 65 degrees and with a specific buffer, and this with the protein is done at a different temperature. So even we know this can work at 65 degrees, although it died after it detects, but it doesn't matter. But we cannot use because the buffers are incompatible. But that is one of the strategies we have, to combine those 2 steps in one. Or even if we get rid out of this step, I mean, just requiring the CRISPR-Cas system is enough. But the problem is that the CRISPR-Cas systems alone have a sensitivity of picomolar, nanomolar of the genetic sequence, which is not that high, specific for not clinically relevant. But for example, with this new CRISPR-Cas711, it could be that we get rid of this step. So that is our strategy, even to combine 2 steps in one. So we will just decrease half the time, or even discarding this step.
Okay, thank you. Yeah.
Thank you so much. We have the opportunity for a very short questions. 1 more. Eden. Okay.
Thank you so much, Armando, for this very nice presentation. This is Eden. Hello.
Hi, Eden.
I hope you are better.
Yeah,
So,
a bit better, yeah.
So my question is, well, if I understood well, some of your systems are based on the kinetics of the fluorescence, right?
Yeah.
So I was wondering if you have explored something related to machine learning to improve the analysis of this kind of kinetics.
Mm-hmm. Yeah. Yeah, what I can say is that we have not explored so far AI, but we have checked some details about the kinetics. So these are enzymes, so we need to use these Michaelis-Menten kinetics. They follow these kinetics. We checked some papers, and they were doing an analysis that these proteins already have a very fast kinetics. So because we also try to do engineering to engineer the protein, so to do mutations in order to increase the kinetics. But these papers that we read, they were saying that basically we cannot, because they are already kind of very high. So I don't know if, after checking those papers, very well done, experimentally also. And also they were saying if you increase a bit the kinetics to almost, to the highest enzyme exists, so the sensitivity won't increase that much, or it will just decrease 100 times, but we need to really go thousands of times, or various order of magnitudes, even lower. So we have not gone into that analysis, but so basically we just went to look for other systems, other CRISPR-Cas systems that could give other opportunities. But yeah, here the kinetics is all about, and also it's very complex because there are kinetics for the cleaving the target DNA, and after this secondary DNA cleavage activity has also other kinetics, etc.
Great. Thank you. Thank you so much for everything.
Thank you very much.
Thank you so much
for the invitation.
Yeah, thank you so much. We just thank again for Professor Armando. So keep it was. So we are going to continue with 2 short talks. So don't go, please. We continue with Dr. Alberto Alejo. Okay. Thank you very much. Yeah. Dr. Alberto Alejo with the talk Advancing Chemiluminescence Biosensing for Harsh Environment, Development of Novel Silicon Carbide Photodetector.